Hello guys, my name is Dr. Varun and here we are with another day with another video of 30 days, 30 most important topic for NEET 2025.
Chalo, let's complete it.
So in this video, we're gonna be dealing with the chapter that is cell division and cell cycle. In this, definitely one question is coming high weightage, 2 to 3 questions can come from this chapter, will come from this chapter, but the one question that is 100% sure that will come, one will be from the Prophase I, and another question higher expectancies from the interphase, that is S phase, G1 phase, G2 phase, G0 phase. Okay, so these two topics we are targeting in this topic, especially the Prophase I, which is most important one. So starting with the Prophase I. So the first in our topic is Prophase I. Now the Prophase I is confusing to the student because a lot of stages are there. Let's try to make it with the diagram and fastly remember the pointers of it. Okay, so now in Prophase I, since it is what? It is a 5-step process. So what are the line-wise steps we have to remember? So the line-wise step, there is a mnemonic for it. What is the mnemonic? That is late zebra passed dancing dog. Okay, so what is this? Late zebra past dancing dog. So L, Z, P, D, D. So the first L is what? Leptotene. The second Z is for zygotine. P is for the pachytine. D is for the diplotene. And next D is for the dikinases. So these are the step wise. Let's just see that what we're gonna see in them. So diagram wise I am making it. So first is the leptotene diagram. Okay, so in the leptotene diagram, what will happen? Your nucleus will start doing what? It will start, nuclear membrane will start disintegrating, membranes on organelles will start disintegrating, chromosome compaction will occur, and chromosomes will ally themselves on one side of the inner nuclear membrane. On the outer side there will be what? Centrosomes present, and it appears something like what? Bouquet. Okay, the bouquet you give it to your girlfriends and boyfriends never get it. We boys should also get it, right? Coming back to the topic, so nuclear envelope disintegrating like this. Okay, so but structure like nucleolus and outside structure, they are still present, they are still started degrading, right? But what is happening? Your centrioles is duplified already, so over here they are present, outside they will start moving. Okay, so they will start moving, whereas inside your chromosome compaction will occur. So compaction means the chromatin is being changed into chromosomes, so like this way. So can you see that this whole structure is something like this way, which is what? How a bouquet look like. So I am enlarging this. Okay, so this is how this stage is called as what? Bouquet stage. So certain points what we have to remember? We have to remember that nuclear membrane disintegrate, or means initiate the disintegration. Next you have to remember what? Cell organelles start disappearing, but they will still be visible in it. Okay, then chromosome will align and form a bouquet stage. So if someone will ask that, leptotene is also called as what? Bouquet stage. Moving to the zygotine. In zygotine what happened? So I am making over here. So in zygotine there is a like what? Now if you see these chromosomes are what? They have extra DNA normally present. So normally the chromosomes is like what? If you normally see, a chromosome will look like this to you. Okay, So in our body, there are 23 pairs of chromosomes. I am just making one chromosome because I can't make that much of chromosomes. So imagine with the respect to 1 multiplied by 23. Okay, so these are the chromosome. This is the one pair of chromosome present. And now what is happening with this one pair of this chromosome? That this chromosome is called as homologous chromosome when they are present together, but during the cell division time they will increase their area, okay, or means increase their DNA content. Okay, so if this is the one chromosome, it will multiply like this. So what actually happened earlier, which was present to be one chromatid in each chromosome, means if it a homologous chromosome means two chromatid only seen, but over here four chromatid scenes. Chromosomes are two only because chromosomes are with respect to primary constrictions. How many constrictions are present? Two. So this is the primary constriction. Okay, but with that, how many arms present? Two arms makes one chromatids. Here what extra arms present? So these are what extra DNA stretches because it has to be divided. Cell is dividing, it need extra DNA. Okay, always remember. So what will happen? That these are what? These are sister chromatid. The ones which I have marked. You will remember this one thing, sister chromatid. Another terminology that you will remember, if you see this and this, if you see this and this, if you see this with respect to this and this with respect to this, they are not similar. Okay, because they are one from mother, one from father. And if you are seeing them, they are chromatids, but they are non-sister chromatids. So this all red color one is what? Non-sister chromatids. Now, this structure looks something different. So, we will give a different name. What would be the name given to them? We will call them bivalents, or we will call them tetrads. Now, this tetrad word is coming because of four chromatids. So, tetrad is what? Four chromatids. Bivalent is coming from the valency. How much extra thing they having? So, 1, that is 2. So, that's why the two extra valencies or chromatids are present to it. I hope these things is understood. Now what will happen during this stage? This stage will initiate the beginning of formation of synoptimal complex. So this become an important statement that during zygote synoptimal complex will form. And if the synoptimal complex will form, then what is this synoptimal complex? Let's see with respect to chromosome only. So the chromosome that we are seeing, in that non-sister chromatids which are closer to each other of the homologous chromosomes, then what will happen? Their chromosomes will chromosomes, they will make what? They will start binding together, okay, with the help of certain proteins and all those things. The closer ones will start going together and start making a bonding with each other. Like the way what? If you went to a new friend place, then you will, like if your new bench partner is there, you will try to talk with him. Okay, so the same thing is over here. So what is forming this synoptimal complex forming? We call that what? Synapse. Synapses. And because of this stage, name changed to what? Synaptotene stage. So zygotene stage is also called as synaptotene stage. Now inside it, if the bivalents are seen, now by this stage still the disintegration is taking place, all those things is there. So homologous chromosome pairing occur and this thing will happen. Chromosome pairing occur. This synoptimal complex that is forming, this join together, more easily or more better feel or seen under microscope during Pashatine. This thing also you have to remember. So this thing I have already told you what is happening inside it. Moving to the next one in the Pashatine. First two stages, that is leptotene and zygotene, is very short lived. Okay, then it followed by the Pashatine. During Pashatine, what will happen? During Pashatine, your recombination nodule will form. So this nodules will be formed in the presence of what? A enzyme called as recombinase enzyme. And if this nodule has been formed, what will happen? Certain structures which are present here will go here and there. So recombination occurring at which stage? Paschertine stage. Okay, so here what will happen? Now if someone will ask like where the homologous where this recombination is occurring, it is occurring between the non-sister chromatids which are undergoing synoptimal complexes. So complexes formation leads to what? Leads to crossing over. Okay, so this will happen. Now by the next stage, that is the diplotin stage, now what will happen? This association, the synaptimal complex, start dissociating, but it completely do not completes. Still it is attached at certain position and it appears like what? X-shaped structure. Imagine that this is the nodule kind of area, which is like first they were like this and then they are separating like this. So they are separating like this. So imagine what is happening. X-shaped structure forms. See this. This is, imagine that this is your chromosome. So chromosome upper one, the finger part is there, this part is there. Okay, chromatids. So it is separating like this. So what happened? X-like structure formed. So this X-like structure is called as what? Chiasmata. So chiasmata formation will be seen where? Chiasmata formation will occur here, which is what? X-shaped appearance of dissociations of synoptimal complex. So means where the synaptical complex portion still left, that is what? Chiasmata. This stage in some of the oocytes, it lasts for years in vertebrates. In vertebrate for years. And if you remember that the oogenesis, it start in birth but is still arrested in the prophase I, which is that phase, diplotin. Important question joining with the sexual reproduction in flowering plants topic. Sorry, sexual reproduction in human reproduction, what I am saying. Okay, so I am also getting little fever, huh? Too much of topics I have to complete. So moving on to the next thing, diagnosis. Diagnosis is called as terminization stage, in which the chiasmata is finally dissociated. That is a terminal stage. Chiasmata will dissociate. Chiasmata dissociation will occur. That's it. Okay, you will remember it. Okay, now we will try to do two questions. One is from the G1 G0 thing and another one is from this topic, okay, which can come to you. So the topic, the question that we're gonna do is what? Let's see. I should have write those questions prior, but it's okay. Number one question from this topic only, that, no, let's just start with this. Context of the topic is what? That the G1 phase, what is happening with the DNA amount, what is happening with the chromosome number. So during the G1 phase, if it is N and if DNA amount is C, okay, the chromosome number is N, means they are haploid or something like that, okay, or I am taking it 2N, okay. Now what will happen during the S phase? Okay, during the S phase, the DNA amount doubles. So always remember, DNA amount doubles means it becomes 2C. But the chromosome is still same. Remember, I made in the diagram that the chromosome will be like this phase. So what happened? Chromosome number is counted with what? Centromeres, but centromeres is still 1. So we will say it 1. Now it will be what? Chromosome number would be what? The chromosome number will be still same, 2N. Okay, moving on to the next one. Next one is your G2 phase, because after that G2 phase come. So in G2 phase, whatever has been double, here also it will continue. Nothing is dividing, and this is still same, that is 2N. Then what will happen? Then your M phase will come. In M phase also, you have to remember certain points, that M phase has what? It has prophase, metaphase, anaphase, telophase, and then there will be what? There will be cytokinesis. So let's just see that what is happening. So during the prophase, still the nuclear identity is there. Okay, one cell only having these things. So 2C will be there. Whereas with respect to M phase also they are still present. So here also it is 2C there. During anaphase they are splitted. Okay, but still the cell is one. So 2C is their amount of DNA that is present. Then in the telophase also two nucleus will be same. Multinucleated condition is there in telophase, but the amount of chromosome inside a plasma membrane boundary is still double. But when the cytokinesis occur, what happens? Two different cells form. Here equal division occur. So means each cell will have C. That is CC. Understood? Now comes with the chromosome number. So in the prophase, there is what one cell is present. The chromosome number is what? 2N. Then in the M phase also the chromosome, like when we are seeing the chromosome, we are seeing with respect to how many centromeres present. Okay, then comes the anaphase. In anaphase, what happened? The chromatids are separated, and if the chromatids are separated, what is happening? Centromeres will appear as double. So here in your anaphase, your chromosome number will become 4N. Okay, then what will happen in the telophase? This again appears same like that. The nucleus is formed, but the chromosomes this will be different for them. And when the next cell will form, at this time the nucleus is divided into two, means the cell is into half. So here what will again 2N 2N. This I have written with respect to your what? Your mitosis. Okay, concept. And on this basis, I am giving you a question. So the question is that what will be the DNA amount of a cell at S and G2 and telophase phase if at G1 phase it has 20 picogram of, means 20 picogram, that's it. So means they have tell that this much amount of DNA is present. So now let's just see that what we have to find. We have to find at the S phase, we have to find at the G2 phase, and we have to find the telophase phase. We know that from this table, when they have not written anything, just written the telophase, means you have to go with respect to mitosis, because in meiosis 1 or 2 word will be written to it. Okay. So at the S phase, we know that everything doubles with respect to G1, because this whole table is with respect to G1. Okay. And G2 phase also it keeps on continuing till telophase it's keep on continuing. So all these phases are what? Continuation phase. Now they have said that given, the given is that G1 phase they have 20 picogram. So can I write 20 picogram at this stage? So they have what? 20 picogram. So it will be doubles to what? 40 picogram. Kg. Very nice. This much. Okay. Then this becomes what? This becomes also 40. This till here will be what? 40 picogram. So this becomes our answer. That is, at this stage we have 40 picogram, 40 picogram, 40 picogram. That's it. One more type of question that we will do, that is based on your divalent and bivalent. Okay. So if during G1 phase 20 chromosomes are present in a diploid cell, how many bivalent and tetravalents present at diplotein stage? Okay, because in diploidine stage only we will see that later on it will divide. Okay, that thing you will make it. If you didn't get it, comment me down. I will share the made out video for that concept also. Similar like the one I have made it here. Okay, the first one. So let's just see that what will happen. Now at G1 phase we have 20 chromosomes. Okay, so let's see that given to us is that 20 chromosomes present at G1. Now we know that after the S phase, so after S phase, DNA material diverges but chromosomes remain same. But the chromosome remain same. But if the chromosome remains same, but what the extra thing forms? We say those structures appear like what? Bivalent or tetravalent. But these chromosome appears as bivalent, or with the term of chromatid we call it tetravalent. Very good. So if the chromosomes are 20, means 20 chromosomes are there like this. So this is what they have said, 20 chromosome in total of the diploid cells. They didn't set the pair. So pair would be what? 10 pairs. So here I am representing one pair. So there are what? Total such 20 pairs present like this. Okay, understanding should be there. Now, how many bivalents will be present? Bivalence means what? One homologous pair represent one bivalent. So one homologous chromosome represent one bivalent, and similarly it represent one tetravalent. How many homologous chromosome present? 20. Okay, so what will happen? 10 homologous chromosome present. We have just calculated on that basis. 10 homologous chromosome present means 10 bivalent and 10 tetravalent present. That's all for this video. If you didn't understand it, let me know that I will make a better explanatory videos in detail about it. Till now, that's all for this video. If you like this video, share it with your friends. Who do you think that this is the most important topic, scrolling topic, and they needed to know? Or maybe you just don't share this video and just keep it with you so that you are able to score good, but they don't. But don't forget to give it a subscribe, because I am making you selfish, but doesn't mean that you will be selfish on me, because I am giving you all of those things. Okay, don't try on me. So that's all for this video. Thank you so much. We will meet soon with another video with 30 days, 30 most important topic for NEET 2025. And on 15th, we gonna be doing the most important topics from each chapter. 3 most important topics from where the paper will come. I will tell you everything decoded. Bye.